Hello, good evening. Welcome on the behalf of Maha AOP. I welcome all this very important session of subspecialty. It's a applied genetics for the general practitioner. When we think of the genetics, which is very super duper subject or only super specialty people handle it, is rarely required. Other than taking history, just taking family history and plotting the pedigree, and that's all right. And thinking of something कुछ तो genetic disease रह सकता है, and we forget to go for the routine cough and cold patients. So what we thought, where this genetics come in picture, applied basics of genetics to the general practitioners, so that they can advise that family to go for the genetic study, as well as whether that patient or future coming generation can be benefited from the genetic therapy. So thanks to first of all Dr. Niman Mohanty sir for agreeing as a chief guest as well as an expert for this session, so that his experience of general practice can guide us through light about this subject. As well, Dr. Snehal and Dr. Parag agreeing and coming as a speaker to this important subject. Thank you very much. Again, I welcome all the dignitaries on the dais, off the dais, all the audience on the YouTube. And I just ask Dr. Amol and Dr. Heman. Dr. Heman Ganguly, the president of Maharashtra AP, and as well as Dr. Amol, the secretary, dynamic secretary of our. So I started our journey great, and I think almost 70%, almost 80% of the year is almost were fulfilled, and at the last quarter of this thing, the Maha AP programs. I think hope it will continue lifelong this. So I first asked Dr. Heman to give his blessing for this session. Thank you.
Thank you, Dr. J. My left hand, Dr. Amol, my right hand. And I do not know about what quarter Dr. J is speaking, but good evening, namaste, Chief Guest Dr. Mohanty sir, and our great esteemed faculty, Dr. Snail Madam, Dr. Parag sir, and all the dignitaries on this dais, as well as the viewers on the Maha AP YouTube channel. One more request, please subscribe our Maha AP YouTube channel. So we want to reach to so many people. This is the 7th episode of Speciality Connect, and hearty congratulations to both Dr. Jay Kumar and Dr. Samir, who has taken the lead, and they have done wonderful work. So hearty congratulations from my side. And as well, our chief guest is there to make the complex science, whatever will be spoken by Dr. Parag and Dr. Snail, to simpler inform to our general pediatrician. And let us proceed to hear all about the complex science, about very complicated subject, genetics. Most of the time in our days, we used to keep that as an optional, and most of the time we used to do that, because almost I cleared my everything in year 1990, so almost 33 years down the line. So that time we used to keep in the option. So let us hear what is this complex material we should know about as a general pediatrician practicing in the this rural area also. So with that, all the best wishes, and thank you, Dr. Jay.
Thank you, Hemant sir. Amol, your words, please. Asa हात नको. तुला काहीतरी बोलावं लागेल. Please.
Thank you, Jay sir. I first of all welcome Dr. Niman Mohanty sir, then Dr. Gangulia sir, Dr. Snail ma'am, and Dr. Parag for accepting this invitation of Maha AP Subspecialty Connect, and they will be talking on genetics today. This is combined initiative of Dr. Jay sir and Dr. Samir Sadawarte sir, the man behind this Maha AP Subspecialty Connect. Without wasting time, I straight away ask Dr. Jay sir to continue the session. Thank you.
Okay. Thank you, Amol. But before start, I hand over to this session to Dr. Samir. I will first take blessing of Dr. Niman Mohanty sir about this topic. Whether it's very, what is the importance of these topics to all the pay attention? Mohanty sir.
Thank you, Jay, and thank you very much for inviting me to this August gathering. Genetics, I think during student days, it sends a chill in our spines. What is this? we thought it is boring, but once you go through, if you go through the Nelson in basic chapters, it's not that difficult. It is only our hesitations which do not steer us there. As you know, like astrophysics, our ancestors also knew something about this, although they did not have exact method mechanism how it functions, but they have some. They had some idea. In 600 BC, they had an idea of Prakriti. Of each individual is different. The treatment plan by them was according to the one's Prakriti and Vikriti, they said. And then came to the Hanuman, homeopathy is the father of homeopathy, Hanuman. Their concept is every individual drug doses do not respond in the same manner of each individual. Treatment they plan according to the even the personality of the individual, they do that plan their treatment. As you know, kapha, bata, pitta, all those things in Ayurveda is there. So that is a genetic predisposition of an individual, how he will behave to a particular regimen. Then came 1991 Human Genome Project. It provided the means of assessing all individuals' Prakriti and Vikriti very clearly. So we have reinforced our ancient idea on this. Without much spending much time, I think we will go ahead with these topics, and in between we may interact.
Yes,
Thank
sir.
you, Jay.
Thank you, Nimen sir. And another.
Dr. Jay sir, Dr. Jay sir, we acknowledge the presence of our CIP EV member, Dr. Pramod Kulkarni sir. He has joined from the US. So we will just ask for 1 or 2 liners from Dr. Pramod Kulkarni sir.
Pramod sir, आहात का?
Yes, it's a pleasure, great pleasure indeed. आणि सगळ्यात पहिलं मला सांगू दे, जेनेटिक्स सारखं इंटरेस्टिंग काही नाही. Absolutely nothing beyond that. सांगतो ना, every third patient in your OPD and practice has some role of genetics in his existence as well as disease and complaints. You just have to have that lookout about it. And I fully agree, and I'm very, very eager to hear the faculties and stalwarts here and the guidance of Dr. Mohanty as well. Thank you so much.
Okay, so genes are in the genes of the should be in the genes of the pediatrics. So I just petition. So I hand over our further session to Samir sir. Please carry forward with and introduce our speakers and start the session at the earliest. Thank you.
We have already seen the mutation in the Samir's name.
Now I have less time, so I will change it later. Let's start with today's topic as a clinical applied genetics. So thanks to Dr. Mohanty sir for coming as a chief guest, and also will be the expert for the today's topic with clinical genetics. Start from with Dr. Snehal Malakmir, and also with us is the Parag Tamankar. So I'll just introduce first Dr. Snehal Malakmir. She is a consultant geneticist and we postgraduate degree in the and fellowship training in the genetics part. And she has her own clinic in Apollo areas and also in the Navi Mumbai with the name Clinic of Genetic Medicine. And her interest is specifically in the genome studies and genome medicines. She is a consultant in the genetics specialty and has receiving all the patients, not only from the pediatricians, doctors, but from general pediatrics, from medicine, from the gynecologist, and from the parent itself. They come and visit her for the genetics specialties expertness. She has been invited many times in the many national and international talk on the subject. Along with us that we have Parag Tamankar, who is having the very special degree, that's a DM in the clinical genetics. And he has completed his fellowship programs and DM programs from Sanjay Gandhi SGPGI. That's a very prestigious institute across India. And he was the first person as a medical geneticist with a DM degrees, started his clinic in Maharashtra, has many public PubMed index journal presentations, and his area of interest is in the clinical genetics, in clinical dysmorphology, inborn errors of metabolism. And he is also the scientist D-type, that's a prestigious scientist program, and he works with the ICMR also. And he's also the treasurer for Indian Academy of Pediatrics Mumbai branch in 2012. So I'm welcoming both Dr. Parag Tamankar and also Dr. Snehal Malakamir Madam. So now whole session, almost up to 1 hour, is on both of you, how to start, how to proceed. And I'm very sure the audience will know a lot of things today about the clinical genetics. So over to Parag and over to Snehal. Thanks. Dr. Parag and Dr. Snehal.
Parag, unmute.
Yeah, thank you,
Yeah.
Dr. Samir. So Dr. Snehal, are you presenting first?
Yeah, anyhow, I'm first, I must say that I'm humbled by the invitation of all the seniors here. And the thing is, we are doing specialty practice in genetics, but I'm sure that all the pediatricians who are listening, they are well acquainted. Only thing is, genetics has a fast pace, and that may be the reason that we are here to give some updates about it. So, Parag, are you presenting first?
You can present, Dr. Snehal, go on.
Okay, I'll share the screen. Is the screen visible?
Yes, madam.
Okay.
Yes, you can make it cup more.
Now?
Yes, full
Yes.
screen now.
Okay. So I am taking one excuse because I have labeled the talk at genomics and applications in pediatrics, because what has happened is COVID has given us a leap. Till now, we were fearing genetics. We were thinking that we do not know, we were not exposed to the knowledge of genetics, but COVID has given us a big leap. We are hearing genome, and that is why actually we are now going into an era of genomics, because we are studying and we are investigating from the perspective of whole genome, whole exome. So we are talking in these terms, and that is why we must be still we are using genetics as a term, because as at our end in India, we have at one end where we are not able to investigate patients even for the basic things, sometimes because of lack of resources or awareness. And at one end we have everything available till whole genome investigations. We have everything available, whatever we want to investigate. So we are into an era where we are using genetics as a term, but still we should know that we are moving into an genomics, and everywhere now the term used is genomics. So that is why I have used it in the, I have labeled the topic like this. So this figure shows nucleotides ATGC, and we are made up of, our genome is made up of combinations of these nucleotides. Almost 99% of human genome is same, meaning the genes and their sequences is same. That is why we are able to study it. So that is there 1%, maybe more than it, that variation, which makes us unique. Each individual is unique. Any change in the sequence, addition, deletion, and now these terms are very easy to use because we are so much used to computation. Earlier it was a little difficult, maybe 10-15 years back, but now we are so used to it that deletion of nucleotides is a mutation, addition, duplication. So these terms are nowadays very day-to-day, I mean, we are using in day-to-day life. So any change in the genome may cause disease. And when we study whole genome, when we unravel the mystery, what Dr. Mohanty was saying, that after 1990s, Human Genome Project was started, and many scientists from multiple countries participated into that investigation, and we sequenced whole genome. Sequencing meaning we now know sequence of nucleotides 1, 2, 3, 4 like this. So we know ATGC, what is the sequence of human genome. And because that sequence is known, we can easily now see the changes in it, which we earlier call it as mutations. This is a central dogma which we learned in school, which we learned in second year microbiology, that we have a gene. We have a gene which is made up of a DNA, which makes up RNA, which is finally made into, after splicing into mRNA. Then there is a protein, and protein is finally folded, like hemoglobin, say, for example. So DNA is made up of exons, introns, exons, introns. Introns are removed by splicing to make a final mRNA. So any change, any problems here because of anything, we get a problematic protein because of which we get disease. So that is a central dogma of life and disease. And then we suddenly know, because now in various meetings, in COVID, in various webinars, we have been connecting to various specialties like pediatric gastroenterology, pediatric neurology, and all the specialties in pediatrics. And we know that somewhere in all these specialties, we are using many of these patients, we are using genetic diseases. So we have been hearing that there are 7,000 known rare diseases. A small portion have approved treatments, maybe more than 500. Some of them are available in India. So what is there at the pediatrician's end, even if it is at a very primary setup in pediatrics, to very highly specialized hospital in pediatrics, what we now need to know is thorough understanding of natural history of the disease, understanding of pathogenesis and targeted treatments, because so much at hand we have now available through artificial intelligence that we can scroll through, we can know guidelines, we can know how to approach this patient. There are so many guidelines available, approaches available. There are tables available. If you say get a patient with hyperammonemia, then we have a table, acidosis, no acidosis. In case of acidosis, ketosis, no ketosis. So the flows are available, everything is available. So now what is remaining then? And now why we still feel that there is something difficult which we can't approach? So somewhere down the line, we have to now start linking ourselves to understand whole natural history of the disease in that particular patient which we are dealing, understanding the pathogenesis, because unless we do this, we are not in a position to explain the disease to parents, explain the prognosis to parents, and whether the treatments are available and how they will benefit to that particular child. So to understand all this flow, we need to understand each disease into that patient in a thorough manner. So sitting in OPD, I have just given 4 pedigrees here. There is one on the left up is, there is a family, parents coming. First child affected, 10 years old male with DMD. Second is 4 years old DMD. Second family comes to us, global developmental delay, maybe multiple investigations done, but very diagnosis, or somebody has done directly, all investigation, genetics also, and there is variant of unknown significance of the report. So there is uncertainty in the diagnosis, and the mother is pregnant now. Then there is third case, very simple, 2-year-old child with thalassemia diagnosed, mother is pregnant. And there is fourth family where there is a child, failure to thrive, speech delay, short stature, so something very abnormal, and we start thinking whether it is genetic, mostly genetic, because this is a very unusual scenario. So we are sitting in the OPD, and now what at pediatricians and what questions parents are throwing on to us. Let us take this example, DMD family, 2 children with DMD. Now, what questions they may be asking, that now even parents know, we once some, and DMD is so easy to diagnose. You get a calf hypertrophy, CPK high, the child is having hours and DMD. And sometimes genetic diagnosis is easily available, so it is done. So now at pediatrician's end, first is whether that DMD has been diagnosed by molecular mutations perfectly, whether that report is positive or negative. Parents are keeping one report on the table of a pediatrician and asking, please explain this report. So that may be the first question. Whether our child will get treatment, because some forms of DMDs now treatment is available even in India. So the next question will be, what will happen to my child, whether my child will get the treatment. Third is whether my child, what is the prognosis of the child? Because parents, if they're educated, they must have read that by 20 years of age, one child will go onto wheelchair. This is having a grave complication, so they may be knowing, but at what rate in our children? Whether whether there is any difference made by adding steroids, what extra care our children need, and how much good quality of life if our children, there is no treatment available, what will happen to quality of life of our children? And because we have 2, whether we can have third child normal. So these may be the questions in the first family. Second is, if all these investigations till genetics we have done and there is no diagnosis, doctor, so what can we do now in our pregnancy? This is thalassemia child. We know now treatment of thalassemia bone marrow transplantation is available at many centers in India. Now what to do? There is a pregnancy. So these are the questions pediatricians may face. And this is the fourth family where there is no diagnosis at all. So what can we do to and start approaching such cases? First question comes to the mind of a pediatrician is to where to begin when we label it as genetic or non-genetic, because genetics has its own taboos. Genetics labeling child or family has its own fears. And if we label the child genetic and it comes negative? So these are all fears in the doctor's minds also. So what are the points which will definitely give us a confidence to tell the family that yes, this is genetic. We need to investigate in step-by-step approach. Some investigations may be costly, and after getting the diagnosis, precise diagnosis, we can definitely give you the prognosis for your affected child. We can definitely guide you for pregnancy options. So first is targeted family history. Targeted I have written because it is very difficult in the busy OPD to get whole history, sometimes seeing such a complicated patient. So very targeted family history, like you know that this is a child of hypotonia, muscular disease, and you are anticipating some muscular neuromuscular problem. So very targeted questions that in last two generations, whether similar symptoms or neonatal death or any problem. So you should ask very targeted questions so that you can get immediate answers in short period of time. 3-generation pedigree is very important if you can draw it. Whether if there are any affected family members, most likely high-risk family to go for genetic disease. If there are any previous abortions, but you don't have any routine investigations positive, so pointing toward genetics. Any abnormal screen test during this current pregnancy of this child, antenatal, postnatal, perinatal, birth course, any abnormalities. Disease in the absence of other known risk factors. You just quickly go to the history, previous investigations or current investigation. You are just not able to point out the finger why there is nothing that I'm not able to find, and still the disease is quite grave. So yes, we need to investigate from genetic point of view. Malformations, intellectual disability, of course, we think of genetics first. And disorders like metabolic disorders, hemoglobinopathies, muscular dystrophies, neuropathies, cardiomyopathies, skeletal dysplasia, hepatobiliary respiratory. There is no doubt that at first visit we can tell that yes, it has a genetic background. The question is whether some patients, many patients we can get positive investigations or positive diagnosis. Few patients, in spite of few investigations, we may not, but still it is highly likely that it is genetics. Sex predilection like DMD, it is seen in males. So males affected in the family or carrier females asking you questions. So it is straight away pointing to genetics. Infertility, unexplained by gynecological causes, and parents are running from this clinic to that clinic, infertility and PGP, implantation, genetic diagnosis, and all sorts of queries. So highly likely that you should have some genetic investigations, and of course, many cancers. So these are the list. This is a list, a quick go through where we can have a definitive approach that we start approaching from a genetic point of view. And in some cases, because now we have set up protocols, we know that this is the MRI picture of this disease and this is the biochemistry. Sometimes we are so confident in telling the child that this is the diagnosis. Like there is a neuroregression in a child, loss of milestones after minor infection. There is a microcephaly you see in the OPD, some dyskinesia, dystonia, and this is an MRI picture. So this is clearly a glutaric aciduria. This is a classic MRI picture of glutaric aciduria, and anybody quickly, even if we don't know, and if we search on internet, will tell that this is glutaric aciduria, patient is suffering. You just sent a biochemistry to confirm and genetic mutation. So sometimes we are so confident, we don't need rocket science to, and very difficult, fear that how to diagnose. This is such a simple picture. This is also simple child that you have some clinical features of delayed milestones, neurological severe impairment or neurological involvement. You have blood biochemistry like acidosis, sometimes hypoglycemia, hyperammonemia plus minus. You have some lactate somewhere, increase, decrease. So you have some biochemistry pointing towards IEM. Then you're sure that this is genetic. MRI showing bilateral hyperintensities. And then you can order easily TMS, blood DCMS. Everybody knows, but this is still, I'm just repeating that these investigations are now cheap. These investigations you can have collected at patient's house at your clinic, at your hospital. Very easy collection, very easy transport. We don't need anything, just call out the lab, they arrange everything. So don't hesitate to do TMS, blood, GCMS, urine immediately in a patient where you suspect IM. Very easy to do. And if there is methylmalonic acid level increased, you straight away go to genetics. So it is so simple for in case of office practice also nowadays. But the problem starts sometimes when there is a wide phenotypical spectrum. When we are sitting in the OPD, we are suspecting, say, a child coming with hepatosplenomegaly, and there is some clue towards Gaucher disease. For as an example, I have taken Gaucher disease. But what happens if there is at both ends of the spectrum, there are some diseases, some features which are not fitting into the disease, some features are fitting into the disease? Then dilemma comes whether it is, I'm feeling that this is genetic, but whether we should order or whether we should tell parents. This happens in all types of diseases where there is a wide phenotypical spectrum. Like at one end, you have an asymptomatic child for few years and then suddenly coming up with anemia and hepatosplenomegaly. At one end, very severe in pregnancy, you have the hydrops fetalis, 2-3 pregnancies lost because of hydrops fetalis, and there is no clue because no investigations done. So there is such a wide spectrum of lysosomal storage disorders, for an example, and that is why which makes us feel that genetics is complicated, but not so. If we try and see, list out the clinical features associated or the progress of the child, as I have said, natural history of the disease, if we document and do one by one investigation, sometimes, many a times, we can know which phenotypical spectrum the child is fitting into and order investigations. For example, in Gaucher disease, it is so easy nowadays that we don't have to do biopsies. We don't have to do invasive diagnosis now, bone marrow or liver biopsies. If you are thinking Gaucher disease, hepatosplenomegaly, anemia, you just take a few blood spots on a filter paper, send it to the company. They will estimate the enzyme levels. If enzyme levels diagnosis are low and fitting into Gaucher disease, they give a mutational analysis and therapy is available as a charity basis after applications. So even in these cases, the investigations nowadays are easily approachable and available. So lysosomal storage disease nowadays, easy workup. When you get skeletal involvement, when you have visceral disease like organomegalies, when you have associated other neurological involvement. So these usually point towards lysosomal storage diseases. If you have sometimes associated very complicated neurologic manifestation, it fits into type 3 or severe lysosomal storage diseases like in Gaucher disease. If they are so severe that there is a pregnancy loss, you start thinking of associated skeletal dysplasia, lysosomal storage disorders. So this is, here we are now, that this is a time where investigations are available. We can easily collect, flowcharts are available, what I was telling earlier, that here we are where we should have thorough understanding of natural history of that particular disease and how it is evolving in that particular patient. If we draw a pedigree and evolution of symptoms timeline, then we will be easily able to do it. It's not that difficult. And understanding of pathogenesis, and then we'll be able to explain targeted treatments in that particular family or a child. For example, if this is, as I have said, that Duchenne muscular dystrophy is so easily diagnosable. We have nowadays combined, because we are seeing delayed development in autistic children, as we all know, that in vaccine charts, we have combined developmental milestones. So suppose a child has delayed motor milestones at 6 to 18 months, 2 to 3. Why I have drawn this 2 to 3 years? Because earlier pickup of DMD and definitive diagnosis, some patients are being taken for treatment even in India on a clinical trial basis, and they're benefiting. Some types of mutations in DMD are treatable now, and many of them at abroad are in clinical trials now, which we are soon expecting in India. So right from when we are documenting growth and development, if there is any clue that there is delayed development at around 2 years, after 2 years, there is a little toe walking over sign or difficulty running, climbing, you go on to do elevated serum creatine kinase. If it is elevated, associated plus or minus cognitive delay, you see a mild calf hypertrophy, you see the child's gait is not good, so, and frequent falls, and it is so easy to diagnose or suspect Duchenne muscular dystrophy. Now, how to confirm that? Straight away nowadays we do genetic investigations. Muscle biopsy is not the first line of investigations. Any invasive biopsies have now reduced, because nowadays we directly collect just blood and send for genetic investigations. So please don't do in diseases where we have a genetic diagnosis definitely available. First, we approach nowadays genetic diagnosis. In cases where we are not getting the diagnosis or the phenotype is not explainable, we are doing and going ahead with muscle biopsies. Or in case genetics diagnosis itself is giving some uncertain answers, then only we are doing invasive investigations. So we have biochemistry investigations like radiological investigations and genetics to make up a large chunk of pediatric diseases can be diagnosed. So what I was making 3 points earlier, that let us understand pathogenesis and natural history of the disease. So what earlier slide showed us a natural history of disease, that motor milestones, slow developmental delay in motor milestones, you get slow calf hypertrophy, a mild cognitive delay, if mother is saying, sometimes hyperactivity in some children. So if that natural history is there, you understand the pathogenesis of the disease, that it is present on X chromosome. So male child coming with these symptoms, of course, it is DMD. And this is a dystrophin gene with 79 exons. This is a dystrophin gene, and I have just shown the figure so that we can understand a little bit of investigations, because we are not going into, I have not gone into details of genetic investigations to keep it simple and a small topic. To take an example, this is a big gene. There are 79 exons at one. These are the ends of the genes. So what happens in DMD is there is multiple exonic deletions. So what we do usually in DMD, wherever there is big part of the gene is missing, that is deletion, we usually apply MLPA. Earlier, we used to do PCR for DMD. Nowadays, we are doing MLPA because we are now studying whole 79 exons. Earlier, we were doing only hotspots or common mutations. And in some cases, if we were not able to find, we used to do MLPA, but now cost has decreased. Therapies are available. So we are directly doing MLPA, because what happens in dystrophin is either there is multiple exonic deletions, say 2 to 8, or there is duplication, say 64, 65, 64, 65. So there is either deletions or duplications. And third type is point mutations. Point meaning at one point in this particular exon, suppose exon number 23, in that exon number 23, there would be a sequence ATGC. And in that ATGC, if one nucleotide is missing, that is point mutation. So there are 3 types of changes in DMD. So how they are studied? Deletions and duplications by MLPA. And if there is point mutation between the exon, if there is any nucleotide change, we study it by sequencing. That we study the sequence of ATGC, we see which nucleotide is missing or any substituted or whatever. And then so there are, in stages, we do MLPA. If MLPA is negative, we go on to do sequencing. That is what happens in mutations. Now, the term mutation, we don't use, we use pathogenic variations. All genetic reports, you will see pathogenic variation as the term. We don't use mutation. I'll tell you the reason why. But this is a simple diagram very commonly used, that how the mutations are there, that either there is single letter change, reverse, deletion, insertions. And if any of the changes in the nucleotides ATGC, whole meaning of the sentence changes. And that is why the protein is so defective that the disease occurs. Why mutations? Because why term was replaced by pathogenic variations? Because as we see in the figure, we are a bunch of human beings, are a bunch of different colors. No color is abnormal. Every color can be normal. So any shape of nose can be normal. And these shapes and these difference and this characterization of an individual is given by their genetic makeup. So this variability is desired because that gives the colorful human community. That doesn't mean that it is abnormal. So what makes it abnormal? So whenever a variation is pathogenic, whenever it is causing a disease, then only we say that it is abnormal. And hence we now use as pathogenic variations. So in the reports, you will read the terms like benign variation, pathogenic variation, likely pathogenic variation. variation of unknown significance. If this variation is benign, meaning there is a variation, but it is not causing the disease for which it is sent. Meaning you are asking some investigation for a suspected disease in a child, some variation is detected, but it is benign. This is not causative of that disease. Likely pathogenic, meaning it is likely that this disease is caused, but maybe scientific evidence is less, or some more investigations are needed, or some family studies are needed. Pathogenic variation, meaning perfectly fitting into the current disease phenotype as explained in the scientific literature. And variant of unknown significance, which is a growing problem, that there is a variation observed after the investigations, but unknown significance. Somehow we are not able to explain, either the phenotype is not fitting, either the genotype is not explained, or the research data is lacking, or the studies in the child are still incomplete, or the family data is incomplete. So these are the terms which we should now be acquainted with, because it is not only for genetic diseases. If you see now infection diseases, COVID, now this coming up swine flus and various viruses, and now we are going to do anyway genomics of viruses and infections also to have a precise therapies. So all these terms will be very commonly used, and we should be aware of it. And these are a few more terms which we should be aware of when we read the reports. This is homozygous, heterozygous, hemizygous, compound heterozygous. Heterozygous, now before that, if say there is a beta globin gene for thalassemia, so it is present at some point on a chromosome. One gene from, so there is one gene responsible for that particular disease. In that gene, one allele comes from father, one from mother. So alleles are different forms of the same gene. So same gene has two alleles. Now, if we go on to this, that's heterozygous, meaning one allele showing somewhere one pathogenic variation. It is called as heterozygous, other is normal. Compound heterozygous, same gene may be, but at different loci, different locus, different nucleotide change. It is compound heterozygous trans. On the same allele at different position, compound heterozygous cis. These terms are used because then you have to explain it to the parent. What do you mean if the report says compound heterozygous? Meaning both the parents may be having that pathogenic variation, but father's is different from the mother's. Homozygous, father and mother, same variation at same locus is homozygous. And it may show, it may point that it is coming from the same community, same ancestor or consanguinity because of inter-community marriages since many generations. So homozygosity can explain that. And double heterozygous, meaning two different alleles completely and different mutations from coming from one parent. So the child shows two mutations, and then we have to go on to do the parental studies. So these are the terms very commonly present in the genetic report, which we must understand. So now again, coming to those four families which were sitting in front of us in the OPDs. If a child comes with DMD, two children, we have to straight away go on to do a molecular diagnosis, confirm DMD, which type of DMD. After approaching, if you do a molecular diagnosis and confirm, if MLPA is negative, please do point mutation by sequencing. Laboratories will guide you. If you can, everybody now is on a phone call away, so anybody can guide you if there is, if other previous investigations come negative. If all the genetic investigations come negative, we are still doing muscle biopsies for DMDs. So very few patients, 2 to 3% patients. And in this child, if the mutation or pathogenic variation is fitting into current treatments available in India, we can refer them. If not available, we have to give them a management plan like steroids, rehabilitation, bone management, fracture management, respiratory management, cardiac management, and monitoring. So this is how the plan goes in this family. And if they want to have a next pregnancy, we have to study mother if she's carrying the same pathogenic variation. And we can give them an option. They can have a prenatal diagnosis in the next pregnancy at 12 weeks by Korean villus biopsy, or they can have pre-implantation genetic diagnosis, meaning a test tube baby can be formed, genetic diagnosis can be done. If the embryo is normal, put it inside. So these options can be discussed by a pediatrician with the family. Now, this is a second family, GDD, Global Developmental Delay, diagnosis VUS. If such child is referred to us very early, mother is into very early pregnancy, 6-8 weeks, and she comes to know that I'm pregnant. What if it repeats? What if my next child becomes GDD or autism? Then what we have to do, we have to go through whole flow of investigations. Directly, we can go ahead with genetic investigation depending on previous investigations or evidence. VUS confirmation needs parental studies, and after this takes almost 4 to 6 weeks in many of the families. So by the time, if they come 6 to 8 weeks, we can definitely give them some option depending on after confirmation by 12 to 14 weeks by CVS. So please don't waste that time. If they come early, refer them directly for prenatal diagnosis very early. We can collect the samples and do the genetic diagnosis. Now, this 2-year-old thalassemia child, of course, we have to do parental investigation. Mother is already pregnant. But what happens to this 2-year-old child? If it is well managed, what we can do is during intrauterine life, everybody knows this maybe, but I'm repeating that in this pregnancy, we are anyway going to do prenatal diagnosis. So if the fetus is normal, we will do HLA typing of the fetus. If HLA is matching with the current affected child, this fetus when born can have a donor, can be a donor for bone marrow to the first affected child. And that is how, pediatrician can convince and cord blood can be stored. So both can be given to this child, and this child can be completely cured if HLA is matching. So both children now, the family will get as normal. And this child will need a complete flow of investigations and the thorough understanding before going ahead with the genetic diagnosis. For an example, if we go, how much time, Dr. Samir? How much time, Dr. Samir?
You carry on. Don't bother about the time, please.
Yeah. No, I'm just.
Because this is a unique opportunity to
10
have
minutes
with us.
more because Parag can take and then you can again come back.
Yeah. So I'm just taking I've just taken 2 cases. One, this is very simple. This child presented at 12 years of age, directly 12 years of age with some autoimmune hepatitis at Apollo Hospital. There was history of TOF, congenital heart disease, and seizures, which were treated with some medications. Now child presenting TOF was operated. Child after seizures or otherwise normal, not a very developmental or nothing, no delay, neurologically normal. Now coming with unexplainable autoimmune hepatitis, so much so that he was being considered for liver transplant. When I went back, if you get a combination like this, you have a TOF child, there is dysmorphism, but I have put it a bar. But there was mild dysmorphism. Father was looking same, but there was dysmorphism which we could make out. So dysmorphic child, congenital heart disease, seizures, please go on to see any biochemical abnormalities and autoimmune hepatitis, so immunological problems. So this triad is different. I have put in a diagnosis actually because anybody can know that congenital heart disease, hypocalcemia, immunological problems, please go on to see Diderot syndrome, 22q11 point deletion. So what next? We can do, if this triad is fitting and in easy diagnosis of 22q11.2, first is in 4 to 5,000 rupees, we can offer a fish. Common deletion can be detected in up to 85% of patients. But what if it comes negative? Then we have to go on to do MLPA. If not, then very few percent of patients, sequencing array, etc. So what are the implications in this child? Because we could diagnose that it is because of this, we treated hypocalcemia, hypoparathyroidism, and immunological problems. The jaundice completely subsided. The child was very well and did not require transplant. So this is what the child could have been diagnosed early, prognosticated, because 22q11.2 do land up in autoimmune problems like autoimmune hepatitis, autoimmune arthritis in later or adolescent life. And definitely seizures could have been prevented if hypocalcemia was detected in childhood age, because in childhood, early childhood we usually do, but in late childhood or a little adolescent, we never do this, think of these causes. So if these are the implications of a definitive diagnosis in such cases. So what to compare, if there are very large deletions and duplications or a large karyotype problem like Down syndrome, Turner, we go on to do a karyotype, or fish like 22q11.2, very classical Williams syndrome, we can do a fish. Microarray also. Nowadays, If the deletions, duplications are in a small part of chromosome, fish specific for that disease can be used. If no fish probe is available for that particular, go on to straight onto microarray. So for smaller deletions, duplications, microarray. If very easily diagnosable conditions by a karyotype, translocations only by a karyotype, especially I've written because of infertile couples and other things, and if we have to do parental investigations for pregnancy, otherwise most of the pediatric cases we may not. And for single gene mutations, sequencing, DNA sequencing. So these are the very, 4 investigations nowadays, so easy to do in any of the, either you do karyotype fish, if it is very specific, we know that it will be useful, or microarray in cases where we know that maybe a microdeletion, microduplication syndrome, like 20 to 1.2 microduplication deletions are also there in some cases, or in intellectual disabilities, or directly go on to do DNA sequencing, exome. Nowadays, people are directly going on to exome, whole exome, because earlier there was a term clinical exome, still we are using it, but the prices almost at par, all laboratories have reduced that we are doing whole exome in most of the cases. This case is quite complicated, but I have taken so that I can explain you everything in this one case, that this is the couple which has approached in the OPD. Mother is 24 years, now pregnant. First pregnancy, MTP was done in view of fetal anomalies like recognizable fetal anomalies on ultrasound, micrognathia, esophageal atresia, lemon-shaped fetal head. Pregnancy was terminated after anomaly scan at 18 to 20 weeks. Now they have come for, obstetrician has referred that because there was earlier MTP, but 2 brothers, one of them had died in 15 years of age because of liver cirrhosis. Other brother, because his brother had problems, was diagnosed a little late by Wilson disease. So Wilson disease were diagnosed in the family by biochemistry. No genetic investigations were done because Wilson disease can be easily diagnosed by biochemistry. There was consanguinity here. So the questions by this couple were what to do, an obstetrician, what to do in this pregnancy, and what will happen to pediatrician when this baby is born. So what happens is, this pregnancy we can do now 7 weeks. So we have ample of time of investigations. So what we can do, if previous DNA, nowadays what we are doing is previous MTP is done, the case is coming to us, we are storing the DNA of previous MTPs at -70 degrees, can be stored for 1-2 years at minimum cost of 500 to 1,000 rupees. And now this DNA can be used, because most of the times during MTPs, parents don't agree with investigations. They're devastated. They don't listen to immediately doctors about any costly investigations. So many of the patients, we are storing DNAs. Now this DNA can be used to see what was the cause of this fetal anomalies. Definitely this and this Wilson disease are not related. So first, if this has any abnormalities, if DNA from the previous pregnancy is not available, we can at least do prenatal diagnosis and see for karyotype or microarray, directly microarray, to see if there are any major chromosomal microdeletion duplicates. So this is for this abnormality. Now these 2 children, her brothers, were Wilson disease. Now Wilson disease is autosomal recessive. So what I explained to that couple is either female will be carrier, or she can be asymptomatic still, because we know that Wilson disease, as I just explained, Gaucher disease has a big spectrum. Even Wilson disease has a spectrum, big spectrum. Some of the adults may be asymptomatic. They may present with neurological manifestations of Wilson disease later in life. So importance of diagnosis in this female first, whether she is carrier or asymptomatic affected person with carrying both mutations. So first, her investigations. If she turns out to be carrier, what will be the implications in the pregnancy? If she is affected, what will be the implication? If she is carrier, she is having only one allele. So, and if husband is having allele, like we do in thalassemia, if she is having only one allele, that is carrier, we have to do husband. If he is carrier, then only we will do prenatal diagnosis. If she is affected, definitely she will be put on a therapy, even if she is asymptomatic. And if they want to do prenatal diagnosis. So what happened is, this brother was not diagnosed and expired, but this brother was very well, because Wilson disease can be completely treated on a therapy. And for information, that is what I was telling earlier, that why we need to understand natural history and pathogenesis, because Indian Society of Gastroenterologists now says that prenatal diagnosis of Wilson disease may not be necessary if family is not willing, because Wilson disease is completely treatable. In case of complications later in life, liver transplant is available. So there is no there is no argument left if we want to terminate such pregnancies, if we can treat them. So the termination of genetic diseases comes if the disease is very severe and untreatable. In treatable disease, you have to give option to the family. So these many things may not be known to pediatricians because we are yet not aware about therapies in genetic diseases. Many Wilson is everybody knows, but many of the genetic therapies we don't know, and we don't give those choices to the family if first child is affected. So this family, the female was carrier. We did NIPT, that is non-invasive pregnancy testing for all the chromosomes. It is a screening test. It was normal for 1 to all 23 chromosomes. So we did not go ahead with prenatal diagnosis for microarray because family was not willing to do. The female turned, we picked up the sample of the brother, mutations were detected, and female was carrier. Now the husband and wife said that because our brother is normal, Wilson disease therapy is available, we don't want to do the prenatal diagnosis. So what was done is we screened for chromosomal abnormalities by screening test called as NIPT. We did not do prenatal diagnosis in this case because they said that even if it is baby's carrier or affected, we want to go ahead with the pregnancy. We just wanted to rule out these abnormalities. So what has this case has depicted is importance of genetic diagnosis in the affected family member as soon as you feel that it is genetic or diagnosis as genetic, so that other family members it is helpful and themselves they can be helped with the therapy. Many pregnancy choices are available to the couple depending on the prognosis of the disease, availability of the treatment, and severity of the disease, and they can be discussed at the parents' choice. And if the disease is severe, if there's say any genetic disease, we had terminated this pregnancy. So first abortion, second abortion, nowadays there are problems of hypothyroidism in the mother, gestational diabetes, multiple complications. In that case, in next pregnancy, do the prenatal diagnosis, and if affected, abortion. Rather than that, we are giving pre-implantation genetic diagnosis a choice that we are detecting mutations in the parents or pathogenic variations, what we call. Then we do an IVF pregnancy, test the embryo. If embryo is normal, put it inside. So all these options definitely can be discussed by a pediatrician. Why not? Why in the obstetrician? Then they are ready to do investigations in the previous affected child. They are ready to take up those prenatal options. They don't run from the door to door. They have a stop at one point where they get explanation of their whole disease. So how to bridge these gaps? Nowadays, as Sir was saying, Pramod Kulkarni was saying, because genetic diagnosis and basis is now available in so many disorders, we are seeing a spectrum of autism, spectrum of epilepsies. So it is to be underlined that relatively common conditions can hide underlying layer diseases like autism. You may be any of these. Epilepsy may be any of these. Many conditions described in past as only clinical ones, such as we used to label that this is just CP, this is just mental retardation, this is just autism, this may be a behavioral problem, psychosis. Now we have definitive classification for it, genetic origin for it, already described. So definitely we should investigate and see for precise cause. And if it is not there, then only we say that unexplained mental deficiency. Before that, we should not label the child like this. Rare disease can be masked by host of other conditions, like we know neonatal sepsis can mask IEM and vice versa, and may lead to misdiagnosis when now managements are available. Over years, infectious disease control, how much achieved, how much remains, because now we have gone through COVID, we have gone through COVID vaccination. So how many vaccines we can prepare if we precisely diagnose infections? Sometimes we can know the burden of the disease in our community, like how much autism is genetic, which we can be prevented, how much autism is non-genetic, where we have to keep continuing our therapies. How much is the immunological disease? How much is the immunogenetic disease? How much is the genetic, so that we can give prognosis? How much of it is just a genetic susceptibility, but may not be full manifestation of a genetic disease? So this can be explained better. Whether can be prevented or handled better, whether there is any prenatal options available, and whether there are any improved methods of diagnosis in genetics. So we at pediatricians, and we need to have understanding of our own data of patients like COVID. We have to know that what genomic pool we have, what screening options like newborn screening in a particular community, how much affording we can do congenital hypothyroidism, CH and 5 disorders, how much we can, in how much population we can apply whole spectrum of IEM. So these policies can only be designed if we understand our own data and prevalence of diseases. We have to build our own resources depending on our social circumstances and needs. We cannot say that the patient is in the periphery and what will happen, no resources, no clinical trials. Definitely somewhere in India available. We are connected now. So we have to build resources. There are many NGOs helping. There is Rett Syndrome Society, Fragile X Society. There are for metabolic disorders also individually. There are parents groups now. There are WhatsApp groups now, DMD groups now. So in India, I'm saying, so we can definitely direct our patients so that they can get help with their like-minded patients and parents. Facilitates, even if concentrated at few apex centers like transplantation, we can reach out, communicate, refer patients early. Crowdfunding is available. Money may not be needed much by the patients to be put in because crowdfunding is available. So we can come together by patient parent groups, support groups, and find out the funding resources, and we can definitely help patients. So there is ray of hope in many of these conditions, either at a preventable end, treatable end, family options end, counseling end, community support, laboratory support, everything is available. We just have to remove this fear that genetic is difficult. No. We have to remove this fear that investigations are costly. No, because the cost has come down. And as I've said, that by doing investigations early, we can put a stop for multiple investigations and multiple visits, and patients, everybody is on the same page, patients will start doing it. So for optimum results, clinicians should balance their opinion based on clinical judgment, utility of that test in that particular disease, strength of particular genetic test. If it comes negative, next what? Routine medical tests should be done, cannot be sidelined. Clinical judgment, routine test cannot be sidelined. We can't just jump onto genetic testing. Importance of them in further management of patients, and definitely clinicians, after balancing all this, they can definitely address parents' concerns about prognosis, pregnancy options, availability of treatment, and most important is consensus guidelines in our type of patients and communities. Pediatricians, all society should come up with their own consensus guidelines that how much in India we can apply to how much, to what extent in multiple communities, multiple states, which mutations are common where, how can we do this? Only can be done if we start doing it, applying it in our day-to-day practice. Thank you. You're not audible, Dr. Samir. Thank you for invitation.
I'm just saying that it was so extensive, elaborative, and very useful. Those who have missed, they can see on the YouTube later. So that's the beauty of having Maha AP with us, where we are doing it live program on YouTube as well as on the our channel. So those who have missed, they can see it again later on. So before wasting our time, I will just call Parag here, and I hope he will talk further more details and will show more number of the patients, and we'll talk about the more of clinical. So over to you, Dr. Parag.
Thank you, Dr. Samir. I would like to thank IIP for giving me this invitation and this opportunity. I will start sharing my screen. Is everybody able to see my screen?
Yes, we are seeing your slide. Yes.
Okay. So I will share some interesting cases. So as Nehal has elaborated on the types of variants, pathogenic variants or mutations. So there are various types of mutations. There could be, so basically in genetics, we have to find the mutation. So once we have a clinical suspicion of a genetic disease, as per what Snehal has said, what are the various criteria for thinking about the genetic diseases, we have to think what mutations can be behind them and how can we diagnose them. So the mutations are basically changes in the sequences. So when we think about the DNA, DNA is like a book. So the chromosomes are the chapters of the book, the genes are the pages of the book, and whatever is written is the genetic code. So there could be problems in the entire chapter of the book or chromosomal abnormalities, or there could be some pages missing that are micro deletions or micro duplication syndromes, or there could be just very small letter changes, a few letters changed here and there, or even a single letter changed. So there could be point mutations, or there could be deletions and duplications. And at certain regions, certain repeats are there. Like for example, in Fragile X, there are CGG repeats. So these repeats are up to some normal extent considered normal. But if they are expanded, if the repeats, number of repeats are increased, then they are abnormal. So basically, a genetic test is analysis of DNA, RNA, chromosome, proteins, and certain metabolites to detect genetic or heritable disease phenotypes for clinical purposes. And genetic testing is now rapidly permeating almost every subspecialty, and hence we have to know what is the actual indication, what is the appropriate use, and the risks and potential for inappropriate use, and various mechanisms of using it in treatment like pharmacogenomics. So when we consider the basic test is a karyotype. So in a karyotype, what we do is we basically look at the chromosomes and the banding of the chromosomes. So the chromosomes are banded in light and dark colors. So that is what is called as GTG banding. So this GTG banding detects an abnormality if the mutation is at least 3 million letters. So if the change is there in 3 million letters or 5 million letters, only then we will be detecting it on karyotype. So for example, Down syndrome, the entire chromosome 21 is extra. So trisomy 21 can be picked up on a karyotype. Then what is FISH? FISH is if we think that the deletion is present, but it is not seen under a microscope. So it is a submicroscopic deletion or submicroscopic duplication. Then we can count the genes under a microscope using probes for as small as 500,000 letters. So this is a diagnosis of a trisomy 21. You can see 3 copies of trisomy, 3 copies of chromosome 21, whereas the 13 number has only 2 copies. So this is a FISH, but you have to know what region to look for, because the probes are very specific and the colors are very specific. So unless you know what you are looking for, we will not be able to pick up on a FISH. Then a very high grade FISH, or you don't know, you think that there is a deletion duplication, but you don't know which area to look for. Then you can use a microarray. So microarray is a counting tool where we count the letters or count the genes onto a chip, computer chip. So basically, we can go even a single letter or even a 10,000 letters. It is much more sensitive than a FISH. And here we can diagnose unknown deletion duplication syndromes other than so it's like a very high resolution FISH where you are looking for variations all over the chromosomes. Then you have sequencing. Here you are seeing how the sequencing looks like. You are seeing different letters, A, C, T, and G, and there are different colors. So the red is the T, black is the G, blue is the C, and green is the A. So basically this sequence is also called as Sanger sequencing. So and we have the latest sequencing technologies. Sanger sequencing is used to sequence only few genes at a time, or you can do few letters at a time, say 1,000 letters or 2,000 letters. But if you want to sequence millions of letters at the same time, then you use something called as next generation sequencing, or NGS. So NGS is like reading the entire book. Sanger is like reading a few paragraphs or a few letters or a few lines. But NGS means reading the entire book cover to cover. So this is how a microarray looks like. This is a sophisticated machine with various parts, and this is how the chip looks like. So gene chip, this is an Affymetrix machine, an Affymetrix gene chip. It approximately the microarray for each, there are various resolutions of the microarray. So for example, if this chip contains 3 lakh probes, then the cost is approximately say around 12,000 rupees. If the gene contains about 6 lakh probes, then the cost is around say 20,000 rupees. So it depends upon how many probes are present on the Affymetrix chip, the cost will vary. So this is a game of resolution. So if you have a much more higher resolution, then the higher the cost. But it is still affordable because you are screening multiple regions at the same time. So there are 2 types of arrays, back array and SNP array. Why it is important to know? Because back array cannot detect the parent of origin of a chromosome. So if you have an imprinting disorder where both chromosomes are derived from a single parent, say from the father or say from the mother, like Angelman or Prader-Willi, then you won't be able to detect uniparental disomy. So SNP array can detect parent of origin of the chromosome. So this is the benefit of using SNP arrays over back arrays. Next generation sequencing is really a marvel, because you can see this is a chip where there are millions of wells in the next generation sequencing, and in each well a single DNA strand is sequenced. Can you believe it? Means It is so small, this well is so small that a single DNA strand will go and fit into it. And as the letters are being added one by one, the camera captures it. The technology is so miniature. So it is nanotechnology. So if we are sequencing the entire genome, so entire genome means 6 billion letters, 6 into 10 raised to 9. If we are sequencing the entire genome, it is called as whole genome sequencing. But if we are interested only in the protein coding exons, then that is called as exome. So the protein coding region of the 6 billion letters is only 35 million. So that is 35 MB data. So that is exon. So it is approximately 1% of the human genome. Then there are various panels available. If you have to sequence, say, only 8,000 genes, then these 8,000 genes are for all the OMIM registered genes. These OMIM registered genes are for all known genes. So you may have many genes for which the function is not known, for which the disease is not known. Then that is the whole exome sequencing. But if you have restricted yourself, see, I have a patient with Pompe disease, very clearly Pompe disease, or lysosomal storage disease, then I can sequence all the lysosomal storage genes. If I have a patient with, say, hepatomegaly and I'm suspecting glycogen storage disease, I know that if I sequence all the glycogen storage genes, I will diagnose approximately 99% sensitivity. So depending upon what your presentation is. But if you have a patient with intellectual disability, there are many more genes which are being discovered almost daily. So then in that case, you will not sequence a few genes, you will sequence the entire exome, because you may be at a point that today you may not get a diagnosis, but after a few years, if we screen the same data again, you may get a diagnosis because that gene may have been discovered, that disease may have been discovered by that time. So basically, we are using the ACMG guidelines for classifying these variants. So they are classified broadly into 3 groups. One is pathogenic, means it is already known that this gene, if there is a spelling change at this position, it causes disease. That means it's a pathogenic variation or likely pathogenic variation. If it is a benign variation, means it is known that it can occur, this change can occur even in normal people. This change is accounting for normal variation in all of us. So which means that 1% of us is unique. We are sharing about 99% of the genes as the same sequence. Only 1% of it we are having unique sequences. So that unique sequences makes us us. So we are having, that is called as polymorphisms. So this is basically very important to know whether it is polymorphic or whether it is pathogenic. And there is a gray zone where we are not able to classify these variants. They are called as variants of uncertain significance. We don't know whether it is normal or abnormal, and that is why we are waiting for more scientific data on that variant, where over a period of time we may be able to discover, or if there are more family members with the same genetic disease, we can then link that genetic phenotype to that new mutation, to that new phenotype, new variation, which we call variant of uncertain significance, and try to classify whether the variant is pathogenic or benign. Depending upon what the spelling change causes to the gene function, you can have either loss of function. For example, you have nonsense mutation, which means that the protein stops at that position. The protein is not, normally protein is around 1,000 letters, amino acid, 1,000 amino acids, but we have a variant by which the protein stops short of, say, 500 letters, 500 amino acids. Then it is called as a premature termination mutation. Or there could be frame shift mutations. Normally the genetic code is read in letters of 3. So if there is single letter change or two letters are changed, then the entire frame will shift. Then there could be problems in the initiation codon, which means in the first letter of every protein is methionine, which is coded by ATG. So if that ATG is affected, if that ATG becomes TTG or AAG, then methionine will be substituted by some other amino acid, and this is called as initiation codon mutation. The common example of loss of function mutations is thalassemia. So the variants which cause thalassemia cause loss of function of the HBB gene or beta globin gene. Then there are gain of function mutations, which means the normal function of the gene is exaggerated. For example, achondroplasia. There is a gene called as FGFR3. The mutation which causes achondroplasia is glycine to arginine change. This glycine to arginine change increases the function of FGFR3, which leads to short stature. Then there is dominant negative mutations. Dominant negative mutations means the new mutation, the mutation produced causes interference in the normal function of the normal gene. So if your patient is heterozygous, there are two copies, one is having the mutation, the other does not have the mutation. If the abnormal copy interferes with the function of the normal copy, then it is called as dominant negative mutations. So the example of dominant negative mutation is osteogenesis imperfecta caused by collagen genes. These collagen molecules are triple helix, and at every third position there is a glycine molecule, which is the smallest amino acid. If this amino acid glycine is substituted by any other amino acid, then it will disrupt even the formation of the triple helix, even the formation of the normal protein, and that's why this is the dominant negative mutations. Then I will come to the examples of cases one by one. So this is the first case, a very common scenario, intellectual disability. But it is not just intellectual disability. In some cases, there may be dysmorphisms. So how is this children different? These children, both of these male brothers, they have dysmorphisms in the form of hypertelorism, which is increased distance between the eyes. There is epicanthic folds, elongated face, large ears, squint, lot of body hair. You can see this small child has almost this entire arm covered with hair, and thick eyebrows. And then their mother had many miscarriages. Their mother had about approximately 5 miscarriages. And then there was, in addition to all this, congenital glaucoma. So this is a child with intellectual disability and facial dysmorphism, and 2 siblings having the same. So what test would you consider? One of the common tests that I would consider in this is a karyotype. So when we do a karyotype, we found that in this boys, both of them, they had an abnormal karyotype. So this is how the karyotype is written: 46 XY common derivative 9. So what is 46 is the total number of chromosomes. So total number of chromosomes is 46. XY is the sex chromosome, and comma given derivative 9. What is derivative 9? Derivative 9 means this long 9 is not derived from only 9. It is made from 2 different chromosomes. That's why it is called as a derivative 9. So on the microscopy, we could find that it was confirmed that there is some proportion of 9 chromosome and 1 chromosome. This is a partial trisomy of 1q. Then we see, when we did a karyotype of the mother, we found that she is a balanced translocation carrier. So this is how a balanced translocation is written: comma XX, comma T1 and 9. So 1 translocated to 9, and these are the break points of the chromosomes, Q41 and P24. So these are where the chromosomal arrangements have occurred. So which means that these 2 children were abnormal, intellectually disabled, and had a lot of this dysmorphism and congenital glaucoma because of the chromosome 9 and chromosome 1 translocation in the mother, and leading to imbalance in the child. So we did a literature review. We found that there were many cases of intellectual disability and glaucoma in relation to chromosome 9p, which means there was some part of chromosome 9 deleted which contained genes of the eyes which caused glaucoma. So this is how gene mapping is done. Gene mapping means we find a critical region for a particular trait or a particular quality by doing review of literature and finding different chromosomal abnormalities. Indeed, this is the critical region which has become haploinsufficient. What is meant by haploinsufficiency? Means if there are 2 copies and 1 copy is deleted, then half a copy is abnormal, and that is called as haploinsufficiency. Case number 2. So this is a child with autism and dysmorphism. So autism has become very common nowadays. The incidence of autism is said to be 1 in 70, and that's why these newer technologies are solving the causes of the autism in many of the families. So in this family also, there was an ongoing pregnancy, and the mother had come to us that she already had a child with autism and developmental delay and dysmorphism, and she wanted to know what is the risk of her baby having the fetus having the abnormality. So we did a karyotype. We didn't find anything in the karyotype. Then we did microarray. In the microarray, we found that there was a deletion of 10q. So the submicroscopic deletion of 10q region, in which we found that many genes were present which led to intellectual disability. So in the fetus, we did the microarray, and the fetus was found to be affected. And you can see the fetus had almost similar facies. This was after the MTP was done, because they did not want a child with disability. So which means there was a submicroscopic translocation in one of the parents which led to this. So here we can find that microarray is a very good tool to diagnose submicroscopic deletion duplications in children with intellectual disability and dysmorphism. So what is the diagnostic yield? So if we screen 100 children with intellectual disability and dysmorphisms, we can get answers in about 20% of cases with microarray and about 5% of cases with karyotype. So out of 100 cases, 5 cases will be abnormal on karyotype, and out of 100 cases, 20 cases may be abnormal if you have not just intellectual disability, but some additional feature like say glaucoma or congenital heart disease or short stature or abnormal facies. Then you can get a diagnostic yield is a much higher. Then coming to various deletion duplication syndromes. So you can see here, there are some of the syndromes you can diagnose just by face reading. So if you have a very typical presentation, this case 3, of course, you can see this is a Williams syndrome child. So this is located on chromosome 7, Q, the Q arm, and you can do a FISH for Williams syndrome, or if the patient is affording, you can do a microarray. In this case 4 and case 5, you can see this is a child with hypotonia, obesity, bitemporal narrowing of the temples and macrocephaly, and this child has Prader-Willi syndrome. So this is a child where there is a deletion of the 15q chromosome from the father's region, from the father's chromosome. And this is a child, you can see this child is a developmentally delayed child, and this child has a wide smile and gesturing like a puppet. So this is happy puppet syndrome or Angelman syndrome. Then this child here, case number 6, you can see there is a Greek helmet-like appearance. This Greek helmet-like appearance comes in Wolff-Fishon syndrome. So if you can see Wolff-Fishon syndrome, you can diagnose sometimes on karyotype. The karyotype showed end of the 4p being missing. So these are all known syndromes, and you can use test appropriately. You can use the FISH test, or you can use the microarray by specifically looking at the features and doing the test. So there is also a diagnosing of imprinting disorders can also be done by doing a PCR test. So in the PCR test, you can diagnose which chromosome is missing. So this is the PCR test. You can see in this test, there are 2 bands. The upper band is the mother's band and the lower band is the father's band. So in the band, you can see the gel image, you can see the mother's band being missing in Angelman syndrome and the father's band being missing in Prader-Willi syndrome. So this is called as a methylation PCR. So in Prader-Willi Angelman, the test of choice is methylation PCR or methylation MLPA. So what is methylation MLPA? In methylation MLPA, we can diagnose not only the deletion, but also the type of deletion. So this is a few videos about the syndromes. So this is a child who had come to us at 5 years. You can see this child is looking almost normal, but to a geneticist, you can make out that this is a Prader-Willi syndrome child. This child was diagnosed at around 5 years of age. It was quite late. By 5 years, the parents had noticed that there was developmental delay, but it was not very severe delay. This child had started speaking late. She had dental dysplasia, but she had good communication. She was not autistic. Whereas if you see a child with Angelman syndrome, a child with Angelman syndrome has much severe, more delay. So this child was also diagnosed at around 4 years of age, very late. And you can see the child has puppet-like appearance, puppet-like gestures, and this inappropriate laughter. So the child keeps on smiling, keeps on laughing all the time. So these are gymnastic seizures. Basically this. So you can make out clinically that these 2 syndromes can be diagnosed. And this is the MLPA, how MLPA looks. So this is normal MLPA where all the probes are present, and when you do an HHA digestion, the methylated copy gets present and the unmethylated copy gets digested. That's why it becomes half. So in a Prader-Willi where you have both the chromosomes derived from the father, the digestion does not take place and both the chromosomes are methylated. In the Prader-Willi deletion, you will see that there is half a copy present, half the copies of these probes are present, and whatever copy is present is a methylated copy from the mother. The father's copy is missing. In Angelman syndrome, you will see that there is deletion of the mother's copy, and whatever copy is present is unmethylated. That's why this is a complete digestion by HHA. So this is how methylation MLPA looks and how it can help in diagnosing and differentiating Prader-Willi and Angelman syndrome. So this is given in most new reports, and you can make out from the graphs that this is a Prader-Willi or this is Angelman. Now another case, this is a child with Duchenne muscular dystrophy. You can see that this is the Pradhan sign. There is a depression in the armpits, and this is the calf hypertrophy. And this is such way the Gower sign can be made out. If you ask the child to stand, the child will climb onto himself, and using his arms to support, he will get up. So this is called as Gower sign. And there is another disorder in which anterior horns of the spinal muscle are involved, anterior horn cells are involved, and that is called as SMA. And in SMA, you will see the typical sign of fasciculation. If you ask the child to open the mouth, you can see that there is rippling of the tongue muscles. So this is fasciculation. So in both of these disorders, you can use MLPA. So in this DMD, you can also use a test called as multiplex PCR, where you see the different exons missing. So in this, you see that a few bands are missing. So that is called as deletion of the exons in multiplex PCR. This is the probes missing in the MLPA of DMD. And this is the probe being extra height in the DMD. So this is a duplication of few exons in DMD. This is the So SMA, what is the pathophysiology of SMA is that there is deletion of exon 7 and exon 8. So if you do an MLPA, the first two are SMN exon 7 and SMN1 exon 8. So both the 7 and 8 are deleted. We also get the copy of exon 7 and exon 8 of SMN2 genes, which is the third and the fourth probe, which is normal. So in certain patients where there is extra copy of SMN2 gene, like say 4 copies or 5 copies of SMN2 gene, then the phenotype is much less severe. So that is why there is difference in the presentation. So if the child presents at stage 0 or at birth or in the first few months of birth, then there are only 2 copies of SMN2 gene. If the child presents at around 2 years, 3 years, then you can make out that the copies are 3 or 4. If the child presents in adulthood, then you can make out the child has 4 or 5 number of SMN2. So the greater the number of SMN2 genes, the less severe is the phenotype. Then these are a family with Fragile X. You can see all the 3 children, they are having dysmorphisms and they are having developmental delay. You can see they are having large teeth, very elongated face, large ears, and hypotonia, and they are very hyperactive. This is the way you diagnose. Initially, we used to use Southern blot. Now, Southern blot is become outdated. We use a test called as TP-PCR. So in the TP-PCR, you use 3 primers, and you can see this is how it looks like a ladder. You see that this is the PCR, which looks like a ladder, and this is a full mutation allele. And this ladder and the full mutation shows that there is expanded repeats. So what is this is a triplet repeat disorder. Normally, normally we have about 50 CGG repeats. If you have 50 to 200, it is called as pre-mutation. And if it is more than 200 repeats, it is called as full mutation or expanded repeats. So these triplet repeat diseases are unique disorders, which is a non-Mendelian inheritance pattern. And few diseases such as Huntington disease. So this is a lady with Huntington disease. You can see, if you ask her to elevate the hand. So this is chorea. So because of the chorea, if you ask her to elevate her hand, raise the hand, she will look as if she is blessing the crowd. So this is called as blessing sign. When you ask her to take out her tongue, it is a jack in the box sign. So this is jack in the box. So this is the repeat of CAG in the Huntington disease gene. So in the HTT gene, there is expansion of repeats. Then this is a girl who in adolescent age came with developmental delay and learning disability and muscle weakness. This sign is called as myotonia. So we ask her to relax and open the hand very fast. Initially, she is able to do. But if you ask her to do it repeatedly, you will see that she's not able to relax it completely. So this is called as myotonia, and this can be even visible on EMG. If you do an EMG, that is called as dive bomber effect. The sound which is made by the EMG machine is called as it looks like a pilot diving in a airplane to drop a bomb. So that is what is called as a dive bomber effect. Both these disorders are triplet repeat disorders, and in the triplet repeat, what we get is expansion. So they may be present in the intron, they may be present in the coding region, they may be present in the untranslated regions. So in Fragile X, you have CGG repeats. In myotonic dystrophy, you have CTG repeats in DMPK gene. In Huntington chorea and spinocerebellar ataxia, you have CAG repeats. So all of these are dominant diseases. The Friedreich's ataxia is a recessive disease, and the repeat is GAA. And then there is an X-linked recessive disease, spinobulbar atrophy, where we have CAG repeats in the coding region. In certain diseases like tuberous sclerosis, you don't need a DNA diagnosis because you have clinical criteria. So if the clinical criteria is satisfied, like in this, you have adenoma sebaceum on the face, then you have Chagrin patch on the abdomen, and you have ungual fibromas on the nails. Then in certain cases, you may have cystic lesions in the kidney, where you have polycystic kidney disease also, because there is a contiguous gene deletion. The TAC2 and PKD1 can be contiguously deleted to cause contiguous deletion syndrome. So you don't require to do a DNA diagnosis. But if you have a family coming to us and they want to have a prenatal in there, they don't want to transmit the disease to the next generation, especially tuberous sclerosis can present sometimes as infantile spasms or West syndrome, where you have severe epilepsy in infancy, and because of that, there could be severe developmental delay and intellectual disability. So in certain families, they don't want to transmit the disease into their next generations, then it is possible to do prenatal, and only in those cases we need to do a DNA diagnosis. And coming to another common scenario, this case we have a 2-year-old girl having regression. So for first 1 year she was normal, then she slowly started to lose all her activities, and she had microcephaly. And in addition to that, she was autistic, and she had these typical hand movements which we saw in the OPD. So in the OPD, you can see that she is continuously doing hand wringing movements. So these hand wringing movements are very typical of a disorder called as Rett syndrome. So the Rett syndrome is one of the most common causes of X-linked mental retardation in females. Like X-linked retardation in males, Fragile X is common. This is one of the very common causes of inherited causes of autism in females, where X chromosome is involved. So this is an X-linked dominant condition wherein the mutation occurs as a new mutation on the mother does not carry the mutation. The father does not carry the mutation. Like in Fragile X, you have the mother transmitting the disease. In this case, the mother does not or the father does not transmit the disease because it's a new mutation. So depending upon there are various types of Rett syndrome. There are various genes for Rett syndrome. The MECP2 gene on the X chromosome is the commonest, but you have FOXG1, CDKL5, TCF4, SHROOM4, SLC35A2, and depending upon the various features, you can have various phenotypes. So for them, what would you do is you would do an exome sequencing. So if you do an MECP2 gene sequencing, you will diagnose about 80 to 90% of Rett syndrome. And in the rest 10%, you can do sequencing of the whole exome to diagnose Rett syndrome. Another child with regression came to us as 2 years, and you can see this sign in OPD. This is called as exaggerated startle. So you clap your hands. This child has repeated episodes of startle. So this child has exaggerated startle, which is a classic feature of Tay-Sachs disease. So when we did the fundus, we could see cherry red spot. So this is a gray matter disease where the cerebral neurons are affected. And there are various gray matter diseases like Tay-Sachs, Sandoff, Gaucher, GM1 gangliosidosis, Sanfilippo disease. So these are all lysosomal storage diseases. And you can diagnose them by doing various enzymes. Like Tay-Sachs, you can diagnose by doing a hexosaminidase assay. Sandoff, you can diagnose by doing hexosaminidase B assay. Gaucher, you can do by doing a glucocerebrosidase assay. Of course, if the patient is affording, you can even do an exome or a panel test for LSDs and diagnose the specific condition. So you can see the Gaucher child has an IV line because this is a patient referred from KM hospital where the patient was receiving enzyme replacement therapy. So this is one of the rare diseases where therapy is available. Whereas in most genetic diseases, there may not be any specific therapy, but there are some where you can really treat and make a difference. Then you have white matter diseases, where the gray matter diseases are involving the cerebral neurons. You have the white matter diseases affecting the myelin sheath. So you have this boy, you can see that this boy is around 6 to 7 years, and the boy started having abnormal behavior and hyperactivity since 1 or 2 years. And the MRI showed very typical white matter lesions of adrenal leukodystrophy. So we did a very long chain fatty acids on a plasma VLCFA analysis, and we found it is very elevated. And when we did the gene sequencing, we found the ABCD1 gene having a mutation. In the early stages, the progression of the disease can be stopped by a bone marrow transplant. So this is one of the rare diseases where a bone marrow transplant can prevent the progression of a disease and save a life. Then you have Krabbe disease, where you can see the child is much younger, is around 7-8 months, and this is an infantile onset leukodystrophy. So the gene is GALC gene, and the enzyme is galactocerebrosidase, which is deficient. Then you have basal ganglia disease. As Nehal has pointed out, this is a child with butyric aciduria. Both children had dystonia, and you can see the second child having a large looking head, that is macrocephaly. And this is a bat wing sign. So the bat wing sign is very typically seen in MRI. So fortunately, these diseases are treatable. So if you treat them with lysine restricted diet, the children can reverse their brain abnormalities and they can lead a normal life. So even if you have a newborn screening, you can detect this on TMS, tandem mass spec, and you can detect in the newborn period and prevent any sequelae. The gene is GCDH gene. You can sequence the gene. It is a small gene. You can do a Sanger sequencing, or if you're not sure about the diagnosis, you can do a panel sequencing or exome sequencing. So we did a study in ICMR where we diagnosed about 30 cases all over India. We found that most of the mutations in Indian population are totally new. So these 2 mutations are all missense mutations. There are very few mutations from Caucasian populations. And that's why when we are doing sequencing, we have to have additional phenotypes such as the biochemical phenotype, which helps in getting a diagnosis, because we may get a lot of VUSs, variants of uncertain significance. And when we want to do a prenatal diagnosis, we have to be very sure that this mutation leads to disease. Then this is a girl with Canavan's disease, one of the rare metabolic disorders which can be diagnosed on a MRS, or a magnetic resonance spectroscopy. So you see the NAA peak. This is the NAA peak in the MRS, or the magnetic resonance spectroscopy. The NAA peak, or the N-acetyl aspartic acid peak, is elevated. So this is an infant with large head and leukodystrophy showing Canavan's disease. So the diagnosis can be made on urine GCMS, where you see NAA elevated in the GCMS, or you can do sequencing of the aspartoacylase ASPA gene, and you can get a diagnosis. A child presenting as spasticity, you can see the scissoring, and this child was labeled as spastic diplegia. The child was mentally normal, but when we did ammonia, the ammonia was elevated and the liver transaminases are elevated, and we did a plasma aminocytogram, we showed elevated arginine. So this is the case of arginemia, which presents like spastic diplegia. We can give a protein restricted diet and other dietary restrictions, and the child can benefit from treatment. Then there are peroxisomal diseases. In addition to the developmental delay, you can get various abnormalities on X-rays. So you can see the X-rays showing the chondrodysplasia. So this is again a biochemical disorder where the peroxisomes are affected, and you can diagnose by doing a very long chain fatty acids or plasmalogen analysis, and you can do a gene sequencing. So this is a skeletal dysplasia with developmental delay and abnormality in the development. Then you have various developmental and epileptic encephalopathies. So you have some children presenting with very severe epilepsy, intractable seizures, and certain genes like sodium channelopathies can be causative. So in this, you have epilepsy and you have various new mutations. So you can see this is a SCNA2 mutation. This is how the exome report looks like. You have exon 28 showing a C dot. So this is how a DNA mutation is written. C dot 538G, which means the A at the 5308 position becomes a G. And this is a heterozygous mutation leading to an autosomal dominant type of phenotype. This was labeled as variant of uncertain significance. But when we did the parental analysis, we found that this was a new mutation because both the parents did not have this mutation, which means this new mutation is likely to be pathogenic because it is a de novo mutation. So de novo mutations occurring in dominant diseases are considered to be pathogenic. So there are various channelopathies which can be diagnosed on this exome sequencing, and these various channels like nicotinic acetyl receptor, sodium channel, GABA receptors or GABA channels, calcium channels or potassium channels. So what is the benefit of knowing? So we know the mechanism of this. Some of the mutations are gain of function mutations, whereas such as sodium, potassium and calcium channels, whereas some of them are loss of function mutations like chloride channels. So if we know what channel is affected, we can give specific therapies. Like for example, in Dravet, the sodium channel is affected. So sodium channel blockers such as lamotrigine, carbamazepine should be avoided, and valproate, topiramate, clobazam, stiripentol and cannabidiol appear beneficial. In potassium channelopathy, you use sodium channel blockers such as carbamazepine and phenytoin. Whereas in glucose transporter defects such as SLC2A1, we give ketogenic diet. In pyridoxine deficient seizures such as ALDH7A1, you use pyridoxine, and in PNPO1 gene mutation, you use pyridoxal 5-phosphate, which are responsive, the seizures are responsive to. In calcium channelopathies, which present such as absent seizures, you use ethosuximide, which reduces T-type currents, and valproate, which are beneficial. In KCNT1 gene epilepsy, you use quinidine. So in certain very specific channelopathies, you use certain drugs. So this is the benefit. This is called as pharmacogenomics. The benefit of using various knowledge of various genes for the benefit of the therapy. Then you have mitochondrial inheritance. So basically, the mitochondrial DNA set is completely different from the DNA set, nuclear DNA set. So nuclear DNA is made up of chromosomes and other genes, whereas the mitochondrial genome is exclusively inherited from the mother and it is present in the mitochondria. It is a circular DNA, and you have two types of inheritance patterns, homoplasmy and heteroplasmy. So in homoplasmy, all the children are affected because all the mitochondria contains mutations, whereas in heteroplasmy, some of the genes are having, some of the mitochondria are having mutations and some of them are not having mutations, and that is what is called as heteroplasmy. So there are various genes for, there are various diseases which are mitochondrial. Some few of them are even associated in the nuclear genome. So there is called as Leigh syndrome. There are two types of Leigh syndromes, mitochondrial Leigh and the nuclear genes causing Leigh syndrome. Then you have exclusive mitochondrial diseases such as MELAS, MRF. MELAS is mitochondrial encephalopathy, lactic acidosis and stroke-like episodes. MRF is myoclonic epilepsy with ragged fibers. LHON is Leber's hereditary optic neuropathy. MNGI is myoneurogenic gastrointestinal encephalopathy. NERP is neuropathy, ataxia, retinitis pigmentosa, and ptosis. So all of these MELAS, MRF, LHON, MNGI, and NERP are pure mitochondrial diseases where mitochondrial DNA is involved. So the tricky part about mitochondria is that not only diagnosing, we not only need to diagnose the presence of the mutation, we also need to diagnose the mutation load. So when we are doing prenatal, for example, we can diagnose that the mutation has transferred or not, but we cannot sometimes able to diagnose the severity of the mutations because the mutation load may be different in different tissues. This needs to be incorporated in the genetic counseling part. So as a geneticist, as we know that many of the diseases are sometimes incurable, sometimes not treatable, and sometimes they are fatal, then in such cases, we need to give the option of prevention of transmission to the future generation of future children by doing a prenatal diagnosis and pre-implantation genetic diagnosis. So pre-implantation genetic diagnosis especially has now become a reality, and you can make out that many of the families which have found a mutation, they can benefit from these technologies. So summarizing, we can diagnose many of the cases nowadays, and if you do extensive genetic testing, at least 40% of the unsolved cases can get solved. Clinical identification is very crucial because you can use a test appropriately. Like for Williams syndrome, you can just do a simple FISH. For doing a Down syndrome, you can just do a simple karyotype instead of doing the whole exome. Or of course, the whole exome can diagnose even the chromosome abnormalities nowadays because you have a tool called as NGS-CNV. Slowly, the microarray may become outdated because NGS is also being able to diagnose microdeletion duplication syndromes nowadays. The knowledge of genetics is complicated by a VOUS. So the variants of uncertain significance and variants with low penetrance are complicating the genetic diagnosis in many patients, and sometimes we need additional information or even additional research into the functional aspects of the mutations. So there is something called as functional assay. So these are various researchers, they want to do reproduce the mutation in the cell line, and they show how the cell line is different from the normal tissue. And this is the area where we can further the molecular biology and find out how different diseases are occurring and how different disease phenotypes are created. Thank you.
Right. Will you please put down the screen?
Yes.
Great. So Dr. Parag, very exclusive way of talking, and we all are happy. Those who are listening here, they got a lot of chunk on the genetics part. I have a few questions as seen in the chat box. Dr. Pramod Kulkarni asked specifically for, should we do for WES, whole exome sequencing, for all undiagnosed cases? My assumption, I will tell you my assumption is like, suppose we found something undiagnosed patient who is like a very sick, not improving on antibiotics, then we think, okay, something might be possible that he might be have genetic disorder, metabolic disorder. We call neurologists, we call people around. They can able to answer some of the patients. And in some cases, they also don't know, then they will send to genetics person. And they always ask for WES, like whole exome sequencing. So a few questions for you is, what exactly is whole exome sequencing and how much time it takes actually? Because that is more interested to know by the people around. And if you don't find any answer on whole exome sequencing, should we go for whole genome sequencing? Again, the whole genome sequencing, the cost will be very high. So Parag and then Snehal, both should ask for this question.
So, so the purpose of genetic testing is to get to a diagnosis. So when we feel that it is genetic, likely to be genetic, like Dr. Samir said, it's a very sick child, and we don't have any explanation why the child is sick. You have you have ruled out a lot of diagnoses like sepsis and etc. Then you can consider doing a whole genome sequencing or whole exome sequencing. The benefit of whole genome sequencing over whole exome sequencing is not much. I think whole exome sequencing is a sufficient diagnosis. The cost of whole exome sequencing is very high. So, and the limitation is that whole exome sequencing can diagnose many cases, but the diagnostic utility is limited to say 40% cases. So if you consider all the cases, if you have 100 such cases, 30 to 40% cases can get solved. And that's why, how you counsel the family. You tell them that even if you spend ₹1 lakh, you may not get a diagnosis in 70 to 60% of cases. Yeah.
And also, why so much time it takes to get the results?
So actually, you know, there are some labs which are doing a test even in say 7 days. The time which is taking is because the tool which is used, the NGS tool which is used, they require some minimum number of samples to be kept. So you have to run the sample in batches of say 30 or 40 or 100, depending upon which tool, which machine they have. So that's why they wait for the samples to get to pool, and then because it's not possible to run one sample and do the entire run for each sample. So that is the reason. So, but there are certain labs where if you give us say 5,000 rupees extra or a premium price, you can get the report within say 2 weeks or so.
So we should go for whole exome sequencing versus whole genome sequencing. Like,
so
can we start
I would
with
say the whole exome sequencing is much better than whole genome sequencing. The benefit is not much. The cost difference is very high between whole exome and whole genome. So I would say for our Indian scenario, I would say whole exome sequencing is good enough.
Okay. Any other question anybody has? They can directly ask here.
May I ask one question,
Yes,
Samir?
sir. Yes, sir.
See, basically narrowing down the possibility of genetic disorder in a child presented to you is the crucial factor to go ahead with the investigation. So the clinical pointers, we have seen many clinical risk factors we have shown in the slide, Snehal showed in the slide. So what is the yield of these clinical factors? Let me know which are the most useful clinical pointers. So let us say consanguinity, then fetal losses, or let us say family history of chronic disorder or mental retardation. Likewise, which are the clinical screening questions which can further give us good clue to go ahead with consideration and investigation of a genetic disorder.
May I, Parag?
Yes,
Yes,
So rather than consanguinity, I mean, we take consanguinity as a risk, but severity of the disease, involvement of multisystemic disease, and a very typical phenotype of that disease, like Gaucher disease, hepatosplenomegaly anemia. So these are the pointers, most of the genetic disorder, multisystemic, severe neurological involvement, any system severe involvement, and then we put on, go on putting high risk factors like consanguinity, previous family members, previous neonatal deaths. So collectively, if each factors goes on to add, then we have a clinical high risk for a genetic disease.
Even if there is no family history, in certain cases, it is important, because if you have ruled out diagnosis like sepsis, infection, etc., then you may need to consider, because certain genes, for example, diabetes insipidus, nephrogenic diabetes insipidus. We had a recent case, I think Dr. Samir may recollect, that from Fortis. So the child had hyponatremia, unexplained hyponatremia, and then they found nephrogenic diabetes insipidus. So in certain cases where you don't have explanation, even if there is no family history, we can do a genetic test.
So in short, meaning severity of the disease, multisystemic involvement, unexplained disease. Like you have thought of all the etiologies and unexplainable. So these are the very high pointers towards genetic disease. And add to it family, consanguinity, previous birth, you go on to increase.
Hello. So Dr. Parag, you very well mentioned about the diagnostic testing to guide the administration of drugs. So how feasible this much in the routine practices? Do you feel it makes some changes? Like we have very sick patients and we on the multiple anticonvulsant drugs. You give some few examples. A very nice one actually. It's eye-opening for me and also for many others who are watching here.
Yes. So if you have a not for all epileptic patients, but only those epileptic patients where we are not responding to therapy. So in those cases, you may tailor your drugs according to the genes which have been obtained on exome sequencing or genetic testing. And then you may really see that some of these reports or some of these results change the way we treat patients.
Samir, can I ask one question? Hello, Samir. Can I ask?
Yes,
sir. Sorry, I was muted. Sorry.
Parag, it was a wonderful talk. Actually, I missed the slide part because I had an emergency call. I just want to know all your correlates of the genetically abnormal disorders on a whole exome sequencing. Like you see some kind of a sequence and clinical correlate and you give it at inadequate relevance or definite relevance or doubtful relevance and whatever. At this point of time, do all those disorders have a definite correlate of your genetic abnormality or genetic sequence? How many of them are having, and with what certainty you can say that this is definitely with this? So what I mean to say, now whole exome sequencing is no more really very costly. It's like a one MRI or a contrast MRI. Cost is as low as that. So many of the patients do go it. So people will start sending it left, right, and center. And you will get those kind of varied reports of varied kind of things. How often do you feel our definite correlate is there, and how many times it can be false positive or false negative?
So, a very good question. So definitely, if you have some clinical pointers which are specific to a genetic disease, the yield is much higher. For example, if you have a hepatosplenomegaly anemia and you're thinking of a chronic hemolytic anemia, then you can say if you do an exome sequencing, you will be able to diagnose 70-80% of cases. But you have cases such as intellectual disability, autism, there you are able to diagnose only 20% of cases. So depending upon the scenario, the diagnostic yield is different. if you get a mutation which is well established by guidelines, by databases, so even you can diagnose this disease in pre-symptomatic stage. For example, you have a family with Huntington disease. You can diagnose juvenile Huntington's even by doing a pre-symptomatic test. So a genetic test is very accurate if you have a very clear-cut mutation. If you don't have a very clear-cut mutation, then that is the area of research. And that research is depending upon various labs or or even certain times, if you have multiple family members affected, you can map that mutation to the various family members having the same disease and come to a conclusion. But genetics is an evolving subject. It is still, a lot of it is in the gray area.
Right. Right. Because now cost of NGS, our whole exam is really coming down. And the people are offering at 12 and 15,000.
Yes,
so
yes.
It's like one MRI. It's a contrast MRI costs sometimes more than that.
Yes.
So it's very easily available.
Yes. The only problem is still most of the labs, they are not able to reduce the turnaround time. So as these tests become more and more approachable, maybe they will reduce the turnaround time also.
Yeah, absolutely. They take about, but actually, really, does it make a difference whether it's 3 weeks or 4 weeks? I mean, your emergency management, apart from these specific syndromes, specific epilepsy syndromes, your emergency management doesn't change. Most of the time is for future counseling or for prevention, because therapeutically, does it really have any role at this point of time?
No, for certain cases, for example, you have, say, a child who is very severely ill and has, for example, end of life decisions, where you have to counsel about what is the long-term prognosis and how much should the family spend. So in such cases, maybe you require to prognosticate very accurately and you require a genetic test. Like if you have a child with SMA, with SMA type 0 or type 1, where of course therapy is available, but it is very expensive. It is approximately 16 crores.
Yeah.
So you have to tell them that you get the money from some source, because it is not still available as a free tool, or you take about end of life decisions. So it is very, very difficult, but still in certain cases you require very fast reporting.
Sir, may I add?
Yes, please.
Sir, few cases in respiratory distress, we have seen that we have detected surfactant receptor or metabolism defects, where it has completely changed the management and prognosis. Sometimes you can give a stop to cocktails. Like we have started some cocktail for IM and patient is improving, but we are in a dilemma and we get negative. You can give a stop to the current treatment and you are sure that we are not dealing with a genetic disease, more we can concentrate on other pediatric causes. So in such cases, definitely such investigations are helping in critical cases.
Actually, that brings us to another Pandora box. How many of the IMs are having specific genetic sequences which can, because IM is a big problem, getting the metabolic disorder on your biochemical test, their sensitivity, specificity, is turnaround time, everything is a problem. And at the end, many times patient land up spending 50-60,000 and still no diagnosis. So how many of these IMs you reliably or with a good specificity can pick up on a whole exam sequencing or maybe clinical sequencing?
So if we go by a specific chart, textbook chart, if there is acidosis, clear-cut indications like hyperammonemia, hypoglycemia, acidosis, ketosis, lactate, where you can put into software algorithms and classify IMs, the yield is very high. In cases where we are suspecting that few pointers on MRI, few pointers on metabolic, even we are not sure whether it is IM we are dealing with or respiratory distress or just a course of NICU which may turn out to be non-genetic. In those cases, the yield is low as 40%. So it depends on the clinical phenotype and data. Definitive pointers, it can reach up to 60, 70, 80%. In low pointers, mild disease, it can go up to 40% and still it can be genetic IM and we are getting negative results. And then all depends on prognosis. And that is why your first question comes that if USS are detected or negatives are detected, then Parag and me, we are not getting now more references for genetic diagnosis. We are getting more of a USS. And I've got 4 USS. So the main first, sir, we have to check is whether we have sent the test requisition form to the laboratory complete. We have to completely draw a 3-generation pedigree on the TRF. We have to completely write the phenotype. The problem is because of the rapid pickup and competitions of the laboratories, they just sometimes share a photocopy or a screenshot of the clinician's notes, where the laboratory person who is putting up the information into the software, we are not sure whether he has put all the phenotypical data, clinical phenotype, biochemistry, MRI, radio diagnosis. So the test requisition form has to be completely filled up. And I have seen if it is completely filled up, if in case there is dysmorphism, the photographs are provided to the laboratory comes back to the present, comes back with the present literature evidence that there have been 2-3 cases more reported, and we are also seeing USS, please correlate. So that sometimes from clinicians end and laboratory end, I think we should give more time to such USS, and we definitely can. And rest of them, it depends on, as Parag said, that evaluating research, family studies, etc. So somewhere I think clinicians also, we are lagging to provide the information to the laboratory, because a lot depends on the filling person, the filling on the laboratory end, also the person who is, sir, why it matters 3 to 4 weeks. They have to literally do the literature search of till the present research. So we should be sure the laboratory scientist is doing it. They have detected the VOS, and if they have completely done the research of recently reported VOS, and if they can get back to us with a publication, and if we can match our phenotype of the child, so we can send the photograph. So there is little after work needed on VOSs, and we can definitely come to a conclusion in, I think 30 to 40% of cases, maybe Parag can add because he's working with a laboratory, but definitely we can add to it. So it is a lot of collaboration still needed in gathering the data and analyzing it. Even if we get VOSs, we can still get the answer.
My questions to Parag and to Snehal. What about the thing we have seen many times, pediatrician just send the samples without involving the genetics person. Like, please do microarray, please do genetic sequence, whole sequencing, exome sequencing, or newborn screening. So multiple things at one time. So do you feel that this number can be reduced by having a proper geneticist in the hospital or in the center? So before sending such tests, it should be mandatory that it should be seen by the genetics. I'm seeing actually in our areas and nowadays, whatever people are seeing. Like,
but not because it is a pediatric meeting, but I must compliment pediatricians. Most of the time they are correct, because they have seen the child at very presentation. If the clinical notes are very correct, pediatricians are really sending wonderful investigations. Problem comes only in milder cases, atypical phenotypes. So we don't mind sending a karyotype in a very typical or a very good dysmorphic child. Intellectual disability, send a microarray. Like Dr. Mohit has said, that you have a clear-cut IEM and GCMS, as I have shown in the slides. You just know that this is methylmalonic acid. Send the exome or send the gene panel. We don't mind it. But where even we are in an uncertain mode that if this is a milder phenotype, the disease and the prognosis is not matching, some evidence is lacking, patient is not having money for doing MRI, which is a definitive investigation in this case. So what to do? In such cases, I think consultation with the geneticist or laboratory will work. But otherwise, sending pediatrician investigations, I mean, that is why we are here, that we are teaching, and they can definitely confidently send the test. We don't mind sending, but only thing is uncertain cases, even we want to learn from pediatricians and clinicians sending it.
No, my question is not like that. My question is, previously, like when 15, 20 years before, we used to send fish karyotyping, irrespective. Suppose somebody looks subnormal. Nowadays, pediatrician directly sending patients asking for whole exome and whole genome sequencing.
Because
that is something wrong.
That is what Dr. Madhuri has asked the question, where what is the difference between array and exome? The recent platforms of whole exome can detect CNVs, that is copy number variations like deletions, small deletions and duplications. So nowadays that distinction is going down. So many of the cases, first people are preferring whole exome. And in cases sometimes very big or micro deletions, duplication syndromes where we are sure that it is not single gene disease, we are preferring microarray. Otherwise, whole exome. So we are moving into whole exome, and soon we will move into whole genome, I think. So that is not always wrong. But definitely, as you said, that if people are not certain, phenotypes are not sure, they can go back and change, because we are detecting 22q11 on the whole exome that there is some deletion in that region, and then we are going back and doing microarray. So sometimes it may not be wrong because of the constraints of the money, because asking patient to do a microarray, and if patient does not turn out, then again asking whole exome. So at the clinician's end, sometimes these judgments may be difficult to cut the cost. So that all depends patient to patient.
Like 25 years before, we used to say newborn screening. Oh my God, why you need it? But nowadays, everybody is sending newborn screening. So do you feel that in next future, maybe in like 2025, 2030, 2040, that whole exome and whole genome will be like just part of patient born and you send the screening?
No, why we are sending newborn screening? Because most of the laboratories after newborn screening positive are doing newborn genome free. So if you're detecting something positive, you get the mutation for free or less cost. And that is why you can start the treatment early. So that is the reason people are sending newborn screening, so that you can, and that the report available is within time before we can go ahead and have mental retardation. So that is one reason. And another reason is not to play with antibiotics, sepsis IM. So better to do newborn screening and then go on to looking. So that may be at pediatricians' end, that may be the good decision points that they are doing it. So, and the cost coming down. So all factors playing together, I think that is a good choice.
Any other questions? I think Dr. Pramod Kulkarni asking, IIP Genetic chapter can consider tele or online advice portal for generalist pediatrician to optimize the yield. Yes, it is definitely should be taken into consideration. Dr. Nimayan Mohanty, sir, you are there. So your thoughts on the today's talk and about the what's the future you feel about the genetics. Dr. Mohanty, sir?
Thank you. We are grateful to Dr. Snehal and Dr. Parag. We have become more educated, or say more aware of this. See the compliments she has given to general pediatricians. It is worth that our pediatrician will take usual proper history, family history, and do a detailed examination of the patient before deciding or to send what investigation to be sent. That is very, very important in such scenario. This dysmorphic features, does it fit to any syndrome? This, if you can try, I think you can get that hint. And selective basis, the investigation should be sent. Not at a random that you do everything, all the battery of investigation. This should not be done. Even they go on to say that what diet the child or the individual will take, these genetic studies will decide, or what exercise the person has to do. All these things, what drugs are going to work, and what dose. These things are coming. I think it's a little much more only on selected hands. Maybe with the geneticists, this should be with the advice of this should be done, but not in a random that we should blindly do such tests. Not much now this time now, so we're not going to discuss more. The talk was excellent, and we want more such sessions where our experts like them can come and educate us. Thank you.
Thank you, sir. Thank you for inviting us.
Thank you, sir.
Thank you, sir. So no other questions there around in chat box. I feel probably we should have a vote of thanks here. So Dr. Jaikumar.
Dr. Samir, you conclude.
Yes, sir. You want to say something, sir? Gangulia, sir?
No, you conclude, sir. Dr. Samir, you conclude.
Okay, I should conclude. Okay. So many thanks to the Maharashtra IIP, Maha IIP, and thanks to President and Secretary for Maha IIP, Dr. Gangulia, sir, and Dr. Pawar, for allowing us as a host for this program, along with Dr. Jaikumar Bhandarkar, sir, who is also the convener, and I am as a EP member to conduct these sessions. Every monthly, we are coming with particular subchapter, and this was our 6th edition. Seventh edition today, for having the genetics expertise with us, with Dr. Snehal and Dr. Parag. It was very excellent way. I feel I need to again read it again or see it again this program, because some of the part really was above my head thinking of what I know. So it was too much things actually. And I'm very thankful to both of them to make the sensitization of the general pediatricians about this subject. And it started with the most basic thing, with the most advanced, completed together. And I'm saying almost 70 of the people are there on the platform right now on this one. And also few of on the YouTube. Many of doctors conveyed to me specifically that we will not able to attend, those who actually attend regularly. So this will be available on the YouTube channel on Maha AP as soon as we finish this program for today. So thank you for coming in a good number and also learning meticulously about today.
Dr. Samir,
Yeah. Yes, sir.
Don't never say allowing us. This is your platform. You're always welcome.
So allowing us, sir, like you selected both of us for this. That's why.
Yes.
Thank you so much, sir, for this.
So not only you, but all Maha APians are invited always on this Maha AP platform. Yeah. They just can share their knowledge. It is for all. Yeah.
And many thanks to Dr. Mohanty, sir, specifically, even at this age as a professor emeritus from the institute, and learning so much things are there. And I have a lot of things learned from you, sir, about this, not only about the general practice or about the clinical methods or acumen. We have to be very serious about the child's health. And I really like you to accept this for today's guest of honor for these programs. So we'll meet again in the next one.
We also thank Chairman Dr. Mahesh Moiti, sir, of the
Samir.
Yes, sir.
Samir, I object to your saying about Dr. Moiti, Dr. Mohanty as at this age. He's just 40 years.
Yeah, that is different.
Very insulting.
I agree, sir.
Objection sustained. Objection sustained.
Thank you, Dr. Mohite, for the compliments. Actually, I beg to add one point here. The information is now the government of India through ICMR is trying to establish centers of excellence in region-wise to do these testings. Maybe Western region we may have at Mumbai. We at MGM we have applied for this to the ICMR. I don't know whether we'll be lucky or not. But other centers like ICMR KM, they are likely to get. In that case, this region people will be very, very benefited. Thank you.
I would also like to give one more pointer. The government of India is reimbursing money up to 50 lakhs for a rare disease. For example, if there is a bone marrow transplant for Gaucher disease, for example, then they can spend up to 50 lakh rupees. It is a one-time expenditure. So even for example, Fabry disease, if they require a renal transplant, then the cost will be taken up by government. So for rare diseases patients, this has come as a boon to them.
So please disseminate this information in form of flyer, in form of format, so it can reach the community of the risk-and people.
Yes, yes.
So we'll appeal once again to everyone. Please subscribe Maha IP on YouTube channel, and you will able to listen and hear and see all the previous programs. We are almost now 350 plus programs on these channels from the start of this year. So thank you once again, and we will meet again in the second of the month on Friday, second week on Friday. And next time it will be topic on the rheumatic and rheumatism and related illnesses. So thank you everyone for attending today's session.
Thank you.
And we'll wind up here. Thank you very much.
Bye.